đón mọi người đến với buổi webinar của ngày hôm nay với cái chủ đề là giải pháp cho carrier screening và quét tích hợp với phân tích CNV. Thì trong buổi này chúng ta sẽ cùng tìm hiểu các giải pháp sàng lọc người mang gen bệnh và quét tích hợp với phân tích CNV. Thì cái việc này giúp nâng cao cái độ chính xác trong phát hiện biến thể và tối ưu hóa cái quy trình xét nghiệm. Thì một lần nữa xin cảm ơn các anh chị đã dành thời gian tham gia cùng với RIVN. Thì đồng hành cùng RIVN hôm nay sẽ có các chuyên gia đến từ Twist Bioscience là tiến sĩ Jim và Andres Pantuni là quản lý phát triển kinh doanh của VarSome, thì sẽ mang đến cái góc nhìn thực tế cho triển khai NGS và diễn giải dữ liệu theo chuẩn của ACMG. Thì RIVN cũng hy vọng rằng nội dung ngày hôm nay sẽ hữu ích cho các anh chị đang làm trong lĩnh vực di truyền sinh sản, bệnh hiếm và genomics lâm sàng. Thì đầu tiên thì em cũng xin được giới thiệu sơ lược về công ty Research Instrument Việt Nam. Thì công ty Research Instrument thì được thành lập vào năm 1989 và vào năm 2022 thì công ty Research Instrument Việt Nam đã chính thức gia nhập và trở thành thành viên của tập đoàn đa quốc gia là Everline. Tính đến nay thì RI cũng đã hoạt động được trên 37 năm, thì chuyên cung cấp các thiết bị và hóa chất trong lĩnh vực life science, cụ thể là mảng sinh học phân tử và protein, tế bào, kháng thể, và mạnh nhất đó là NGS giải trình tự cho các giải pháp về y sinh học liên quan đến giải trình tự gen. Thì RI hiện tại là có mặt ở 4 quốc gia là Việt Nam, Thái Lan, Singapore và Malaysia, và có trụ sở chính ở Singapore. Và đối với Việt Nam thì có 2 văn phòng là Hồ Chí Minh và Hà Nội. Thì cái việc này sẽ giúp cho RI có thể hỗ trợ khách hàng tốt hơn trong cái việc mà chạy máy và những cái xây dựng các giải pháp đồng hành. Thì nói đến các giải pháp giải trình tự NGS mà RIVN cung cấp thì sẽ có là giải trình tự gen của thế hệ thứ 2 và thế hệ thứ 3 là giải đoạn ngắn và giải đoạn dài. Thì bên cạnh cái giải pháp hệ máy NCI thì tụi em cung cấp full giải pháp và từ cái hóa chất chuẩn bị thư viện, hóa chất giải trình tự, hệ máy cho đến cái phân tích. Thì cái nội dung của cái cuộc webinar ngày hôm nay là sẽ sử dụng cái hóa chất của hãng Twist Bioscience cho cái bệnh hiếm carrier screening cho West và chạy trên cái hệ MGI, và sau đó là sẽ phân tích bằng và VarSome. Thì bên cạnh Twist thì tụi em cũng có những cái giải pháp khác về IVD như là của hãng BGI chuyên về ung thư, NIPT, hay là những cái Genomics về ung thư, và hãng Di Công cho cái sàng lọc phôi. Thì bên cạnh đó thì tụi em cũng có cung cấp các giải pháp về protein và tế bào như là hãng dừa sinh học Citiva chuyên về các hạt bit để mà tách chiết tinh sạch protein và DNA, nanostring đo cường độ biểu hiện gen và kháng thể của protein tech hay cell signaling, và ELISA kỹ thuật số của hãng Quanteric. Thì em cũng xin được phép cho giới thiệu Jim đến từ Twist Bioscience sẽ tiếp tục cái cuộc webinar ngày hôm nay. Hi Jim, you can share your screen and start the webinar.
Thank you, Ms. Fan. Let me share my screen right now. Can you see my screen okay?
Yes, I can see your screen.
Thanks, Ms. Fan. Yes, I think let's get started. So thanks everyone for taking time this afternoon to join this joint webinar from Twist Bioscience and VarSome, right? So my name is Jim. I'm a senior field application scientist from Twist Bioscience. So very happy today to share with you some of our latest NGS solutions, right? That is really focused on, using the whole exome sequencing technology and incorporating copy number variation detection for rare disease research. And also towards the end, I will share some information on how Twist can help support carrier screening as well through our custom panel design and synthesis platforms. So for rare disease, I really wanted to start by talking about the burden of rare disease, right? So today there are in fact more than 8,000 over rare diseases that has been identified globally, and many individuals, many people who have been identified as having the rare disease they unfortunately only 5% of the conditions are currently having any form of treatment available, right? Majority of the people, once we are diagnosed with a rare disease, there is effectively no therapy or no cures available. And so we really wanted to understand the genetic basis for rare diseases, because 80% of these diseases, they have a genetic aberration, right? Genetic basis for the cause of the rare disease. And 75% of these diseases, they actually present themselves through a wide range of different symptoms, right? It could be neurological, it could be physical or intellectual. So this makes it very hard to diagnose this rare disease accurately. And many of these diseases, unfortunately, they affect children, right, under the age of 5. And 1/3 of these children, unfortunately, they will die right before the age of 5 due to the severity of the symptoms. So this obviously has a massive impact on millions of families worldwide, and it really is a reminder for us that it is really important to focus on the research to understand the disease and to help improve diagnostics for rare diseases. So from the map on the right, we see that actually the Asia Pacific region where we are at has the highest rare disease burden in the whole world, right? There are over 250 million people living with a rare disease in Asia Pacific, because our part of the world account for more than 60% of the world population. So I think it's a major, big public health challenge in many countries, and it's not just for individuals and families, but also for global healthcare systems as well. So one of the biggest challenge is that because it is rare, so it's not easy to diagnose. So it's a long and complex process, and the patient and the families can have a very long and difficult path to obtain an accurate diagnosis, because the lack of knowledge means many doctors have limited exposure and they can sometimes not recognize the symptoms correctly, and they have this can lead to bias in the diagnostics. So doctors can lean towards more common explanations for the symptoms, right, and overlook that there is actually a rare disease, right, in the patient. So another problem is rare disease can present themselves in different ways. So the same disease can show different symptoms from one patient to another. So for example, the classic example is cystic fibrosis, right, which can be caused by over 2,000 different mutations. As a result, that same disease, different genetic profile can present different phenotype in different people. Some people just have, very mild respiratory symptoms. Some people have very severe lung disease, right? And we have some other issues as well, access to testing centers and correct diagnostic tools. And these are some other challenges. So it's not a straightforward path, and is, a lot of work remains to be done to improve the diagnostic efficiency, to help to improve the patient outcomes. So NGS right now is one of the most important and promising tools for rare disease research to help to improve the diagnostics. So this is how the overall workflow looks like. It often includes synthetic DNA controls on the left here. These are engineered reference materials, reference DNA, that comes with non-variant allele frequencies, non-mutation frequencies, to help us validate the NGS assay and to perform quality control of the NGS assay. So we have library preparation to process the sample. The patient sample could be DNA, could be RNA, could be methylated DNA. We process them for sequencing via a very streamlined, now I think it's really streamlined. We have very specialized technologies to process these samples efficiently and quickly. And to look at specific disease genes, right, we use target enrichment. So this could be a custom set of genes for a certain disease, or it could be the whole full coding using the exome, right, to capture full coding regions. And more recently, also DNA methylation signatures are being used for rare disease research as well. So right now, I think NGS is considered a mature technology. We already have many competitive and new platforms on the market. Previously, Illumina was very dominant in this space, but now we are seeing many more cost-effective platform, and also sometimes even higher sequencing accuracy, right? MGI, Element Bioscience, Ultima, and long-read platforms, Oxford Nanopore, PacBio. Some of these new platforms even start to incorporate multi-omics capabilities, such as spatial transcriptomics and even proteomics, right? So this is a very fast-moving, I think, industry, and there are, it's not just at research front, but also for clinical diagnostic use as well, right? Post-sequencing, we also have many software vendors such as VarSome to offer that complete sample to reporting workflow for the rare disease, screening workflows. So you may be thinking, so with the new sequencing platforms, with sequencing costs coming down, why isn't everyone doing whole genome sequencing, right? Since it's kind of getting more and more cost effective, we start hearing like less than $100 to sequence a whole genome. I think while the price is falling very quickly, it is still relatively expensive because of the size of the human genome at 3 billion base pairs is still a very big space that we try to sequence. By using targeted sequencing, as we show here in this figure, right, we only focus the sequencing resources on specific regions of interest. So this means that firstly, the coverage that we can get will be higher sequencing coverage shown here that can help us increase the sequencing accuracy and increase confidence in the mutations that we identify. This is one thing. Second thing, it also means that we can process more samples in a sequencing run, which can help us reduce the sequencing costs, right? So there is still a lot of value in doing targeted NGS, and I have not even touched on the costs associated with data storage and also data transfer, which can become very significant for whole genome sequencing because, that number add up very quickly with more samples being sequenced, and those data needs to be stored for a long period of time, right? We're looking at 10 to 20 years of the data storage duration, and all that costs will add up significantly over time. So when we look at targeted enrichment, one of the, in fact, most powerful approach, right, to perform targeted enrichment for rare disease is to perform whole exome sequencing, because with just 1% of the genome, by sequencing just 1%, we are in fact able to identify up to 80 to 85% of the disease-causing mutations in the genome, right? So this is a very powerful approach for rare disease diagnostics. There are many different strategies towards whole exome sequencing. Different platforms, different panels have different definitions of what an exome panel should be or should look like. So at Twist, I think we focus on the clinical utility of the exome panel. What this means is that, for any clinically relevant gene, so this could be genes that are recorded in the genetic testing registries or genes which are highly cited in the literature, we focus on full, gene coverage. All the coding exons are being covered. There are still quite a number of genes in the human genome which are poorly annotated. Some of them may not even be experimentally validated to exist. Some of them, we just simply, do not know what is the function. They have not been implicated in any disease. We kind of assign a lower priority to those genes, meaning if there are some regions which are, associated with higher off-target or, high risk of off-target capture for those genes, deprioritize that, and we actually filter out the probes in those regions. What we're really trying to do here is to use a very, I think, careful curation of the panel to ensure that the clinical utility is highest. At the same time, the panel is not too big. And at the same time, the performance is best in class, right? Because we only sequence the specific regions that we want with minimal, what we call off-target risks. And end up, we capture regions of the genome, which is not, coding exon, for example, which just wastes the sequencing data. So this is at the design front. The other front, we really want to emphasize to overcome some of the challenges in whole exome sequencing is the quality of the reagent itself, the quality of the panel itself. I think Twist is in a very unique position to solve this question because of our silicon-based DNA synthesis platform. Right? So this platform allows us to miniaturize DNA synthesis in what we call nanowells. They are being shown here, right? Each of these are the nanowells. In each nanowells, there are multiple micro droplets, and the DNA synthesis takes place on this silicon chip, and there are about close to 10,000 of the nanowells on this chip, and allows up to 1 million oligos to be synthesized in one run. So this is around 1,000 times higher than standard synthesis methods. So because the synthesis is being miniaturized in such tiny droplets, right? We can control the synthesis condition, the temperature, the reagent concentration. It can be controlled much more effectively, right? Efficiently, and this increases the uniformity and the accuracy of synthesis. So we can see here with the Twist panel, you get a much tighter distribution in terms of oligo copy numbers. Is very tight across the whole range of GC content. Now, why is this important? This is important because all these oligos being synthesized, synthetic DNA oligos, are what we use in the exome panel to capture the regions of interest. And having a very uniform oligo panel means that the target DNA that is being captured, we can obtain a very uniform capture. What we're trying to do here is we want to minimize any over sequencing and under sequencing, because all these are wasted sequencing data, which in the end leads to higher sequencing costs. So by using the Twist exome panel synthesized in this unique way, we get much more uniform coverage so that we can reduce the sequencing resources that we need. And this is, what we're doing, in the background to synthesize, right? And this has in fact been tested, right, and validated in an independent study that was published from a group in Netherlands. So they wanted to understand, right, the use of exome panels for clinical exome sequencing. There are still some challenges. The challenges that sometimes can affect our ability to call mutations and copy number variants sensitively. So because some of the regions in the genome, what we call regions with high GC content or high AT content, those still tend to be not as captured effectively for whole exome. And we have some regions not being captured well. This can affect not just mutation, single nucleotide variant detection, but also for copy number variation detection. So the goal was to kind of compare different commercial panels and also compare them with whole exome sequencing to see which one could provide the best coverage and also the mutation calling sensitivity. So here they compared, the exome from Agilent with Twist, with short read and long read whole genome sequencing, right? And the result is showing here the coverage percent of the target which meet 20x coverage. And here you can see, like, in this case, right, Twist exome performance, right, is almost comparable to short read whole genome sequencing, while you're only targeting a very small portion of the genome, right, around 1% of the genome, 1 to 2%, but being able to cover all the RefSeq and ensemble coding region in a very equivalent way, right? So this is important, right, to make sure there is minimal underrepresentation of target region that may harbor important mutation that could be driving rare disease. So the other important metric is what we call the coverage uniformity, right? We can cover all regions, right? But not all regions may be covered equally well. Some may be over sequenced, some may be under sequenced. So they use a metric which they to use to calculate like the kind of uniformity of the capture. And so this is, you know, from Twist, we can see that likewise, right, the uniformity, the evenness of the capture is also, you know, significantly better than other exome panel options, somewhat almost catching up to whole genome sequencing levels. So I think this is really going down to how the panel was designed, how we designed the probes, the placement of the probes, and also how the probes were being synthesized, right? At these exome panels from Twist for specific regions with, you know, high or low GC content, the panel is actually boosted, right, to balance the coverage in those challenging regions. And we see that, you know, in an actual validated study, how it actually helps. So this is all, you know, regards to the capture performance. They did went on to evaluate the performance of the panel for CNV and SNP calling, and they found that, you know, you can reliably call the SNPs at 70x coverage, right? Whereas for 70x coverage is enough for them to perform reliable SNP and CNV detection, whereas for other workflows, it requires higher coverage, 100x or even higher. So in a way, you know, we are seeing like significant saving costs because of the higher uniformity. It means that you just need to sequence not so deeply in order to obtain enough information to call out these variants confidently, whether it's SNP or copy number variation. So I talk about copy number variation. So it is possible to do copy number variation with exome sequencing. However, the limitation is that exome panels, they have significant gaps in the whole genome, right? The exons here, they are like shown here in red. So they are spaced out from one another, and in between genes, we have huge gaps. It could be introns, it could be intergenic regions. And so any copy number variation events at these regions, basically we cannot detect using a whole exome panel because the coverage is 0. So it's only 1% of the genome. So the gaps are huge. That gap contains 99% of the human genome. So it's not possible to perform comprehensive copy number variation using exome sequencing. And copy number variation is, in fact, very important kind of biomarker, not just for rare disease. A lot of neurological disorder, and in fact, even cancers are very, you know, associated with certain CNV biomarkers. So what we have done is that we have actually designed different spike-in panels at different resolutions to complement the Twist exome panel in order to provide us that full genome coverage to call out CNVs across the entire genome. So this is what is being represented, right? The gaps shown here in red are the intronic or intergenic regions that we get from a whole exome sequencing approach, right? So these panels, we call 25 kb, 50 kb, 100 kb, represent different resolution. It depends on our sequencing needs, what, you know, the goals of the study. We may be looking at detecting copy number variation at different resolutions. What this means is the spacing between the individual probes. So the 25 kb panel actually has the highest resolution in our CNV detection capabilities, and it really fills up the gaps and provide that whole genome coverage. So what this looks like after we perform the capture. So this is a spike-in panel, which means that the exact same workflow we perform with exome sequencing, we spike in the CNV panel, one assay, one sequencing run, we can obtain full exonic information as well as whole genome CNV detection capabilities. And at the end of the day, after sequencing, right, this is what we get in terms of the output, is that we get, you know, the single base pair resolution in terms of our ability to detect SNPs, and also via so each of these dots here represent each of the SNP that we include, right, in in the spike-in panel so that we are able to detect any CNV events via the coverage that is obtained after sequencing. So in this example here is just trying to illustrate how sequencing coverage information can be translated very reliably into CNV level information using the exo panel with the spike-in panel. So this is what I want to share for I think I'm almost out of time here, but that was for the fixed panel, and we understand that the off-the-shelf panels may not always provide the specific targets that we need for the specific study. And this is in the context of carrier screening. For example, different populations will have a different set of genes that is implicated in the carrier status of different populations. And that can be different because of the genetic pickup of different populations, and also the disease prevalence in different regions can be different. In that case, right, we really need to adopt a more customized approach in our sequencing and in our the panel that we use. So I just want to quickly share this design, build, test, optimize process that is specifically tailored for specific workflow, right? So we not only deliver the panel for the specific targets, but we also ensure that because it's a custom panel, so we're not able to predict the performance, but this workflow helps to kind of refine any custom panel until it meets your specific needs, and we do not compromise on performance, right? We build it using the same silicon-based oligo platform that I just shared, using our nanowell-based system to ensure the oligo is uniform. So this goes through a round of testing in, I would say, 90-95% of the time, the panel works as expected. In the event that we require the panel to meet certain specifications or metrics, for example, we need specific coverage at certain genes, we can optimize and fine-tune that design very easily. And you can work with our bioinformatics team or our field application scientist team in the field to help to kind of obtain the best possible custom panel design. And that approach is the exact same way in which we help this particular study to really showcase the importance of panel customization for carrier screening. So in this study, the authors, right, based in China, they wanted to address a specific clinical gap in the standard ACMG recommended gene panel for carrier screening. So the genes, so ACMG recommends 113 gene panel for carrier screening. This is based on European populations. It may not be suitable to capture the carrier burden in Asian populations. So to address this, right, the authors, they designed a Twist custom panel, and they target 334 genes because they wanted to understand, right, if we use an expanded carrier screening panel, what is the impact on the carrier screening rate, right? Instead of just relying on what ACMG recommends, they expanded it to 334 based on genes that are significant in Asian populations. So this includes genes like G6PD, thalassemia, which is not really being prioritized in the European populations. So they apply this panel to over 2,000 individuals, over 2,000 healthy people. They don't have any clinical phenotype, and use the panel for for carrier screening, identifying the variant and interpreting the variant and calculating the carrier rates per gene. So it's very interesting because they found that more than 60% of the people, and these are healthy individuals, they carry at least one disease-causing variant. So it's nearly 2 out of 3 in healthy people being screened that were the carriers of some disease. And the overall the at-risk couple rate 11.76%. So 1 in 9 couples in this cohort, they face some reproductive risk that standard screening may miss. So I think, yes, they did some more analysis, which is, I think, quite interesting because not all genes contribute equally to reproductive risk. Some genes more, some genes less. But when they set a cutoff of 1 out of 1,000 for the carrier rate cutoff, they found that this smaller set of genes can still capture over 90% of the cumulative carrier risk. So this is an important finding because it means that for this particular population, right, they can have an evidence-based threshold to build a more cost-effective and more population-specific carrier screening panel. And there are some interesting genes here, GJB2 for hearing loss, and then alpha thalassemia, HBA1, HBB, thalassemia genes. So these are genes that simply cannot be detected by using a panel that is very focused on Western centric populations. All right, with that, I think I'm almost up on time. So thank you for your attention, and we'll be happy to address any questions at the end of this webinar session.
Yeah, thank you, Jim. We will have the Q&A question after the meeting, and then we have the card with Jim. And I hope that the participant can join into the mini game, and RIVN also have the small gift for the people who can answer the right question. Next, we would like to invite Andres Pantunis, the Business Development Managers from VarSome, can present how we can analyze the data for the NGS Park. Hi, Andres, you can share your screen and start.
Of course. Do you see my screen? Do you see my screen now?
Yes, I can see your screen.
Perfect. Well, thank you. Thank you very much, everyone, for attending. Thank you, Jim, for your presentation. I'm Andres Benito. I'm a business development manager in VarSome as a feeder, and I work mainly for the APAC region. So today I will be talking about our solution, VarSome, and how this can help streamline the secondary and tertiary analysis. So the first thing I would like to talk about is our goal as a company. So basically what we try to do with our solutions is to enable our users to find, share, and use the most comprehensive human genome data that there is, and to collaborate to improve the health and lives around the world. Here you can see in the map the different regions in which we have VarSome users. So we have now more than 500,000 users all around the world. So we are pretty known. And the first thing I'd like to share with you is the free platform that we have. This is where we have most of our users. And basically, what we try to do with this is to help the users get to the information for specific variants, because as you know, there's many different data sources. So what we try to do is we try to harmonize and integrate all of these different data sources into one specific platform to make it easier for the clinicians and for our users to get the specific information and the annotation for the variants. So here in varsome.com, users can input specific variants, and we will provide the annotation and classification. And here you can see a quick snapshot of some of the information. And we will also provide the classification based on the ACMG guidelines and AMP guidelines. Apart from the free version, we have other solutions. First of all, we have VarSome Premium, which enables you to integrate additional data sources compared to the free version, and it has also unlimited queries. Then we have VarSome Clinical, which is our main product. In this platform, users can upload either FASTQ or VCF data, and they can basically filter through all of the variants in their samples and generate a report. I will take a few minutes afterwards to show a little bit more about VarSome Clinical. And then we also have VarSome API for institutions and labs that have a bioinformatics team, and basically they want to integrate all of our knowledge base into their pipeline. So these are the different solutions. As I was saying, here we have VarSome Premium, in which you can see different information, including publications, community contributions, the germline and somatic classification, in silico predictors, etc. And this is also included in VarSome Clinical, as I will show now. So here we have some of the most important features. So we have more than 140 sources integrated into the platform, over 4 million scientific publications that we will link to the specific genes and variants that are in the samples that our users upload, and also in VarSome Premium. And then we also provide the germline and somatic classification, and we show which of the rules have been applied. And we also enable our users to modify this if they have additional information. Apart from this, we also have a team of manual curators who manually review the publications we link to at the variant and gene level, to make sure that the publications are correctly linked. So here we have a few more snapshots, and we also annotate novel variants. So here we have an example in which we see that a VarSome curator has curated a specific variant. So what we try to do, as I said, and what is our mission here, is to basically try to harmonize all of the information there is out there from the different data sources into one specific platform to make it easier and faster to our users to analyze the specific variants. So in terms of our solution, we are compatible with FASTQs and VCF files, so users can upload both type of files. From FASTQ, we support Illumina, MGI, and Element. And from VCF, we support any standard VCF. In terms of the pipelines that we can support, we support germline and somatic use cases. In germline, we perform solo analysis, carrier risk for couples, trio analysis, and cohort. And then in somatic, tumor only and tumor normal. And in terms of the variants that can be analyzed in VarSome, we support SNPs and small indels, and also short tandem repeats, structural variants, CNVs that we will also discuss in more detail. We also have information about TMB and MSI, and recently we have just released fusion annotation from RNA. Basically, we have, as I said, we try to provide answers for our users about these specific variants, and we have a very simple user interface. So as soon as you sign up in VarSome Clinical, you will see that it's quite easy to move around the platform and analyze your own samples. We also have some filters that you can create and customize, and basically enable you to filter throughout anything. I will show this in more detail. And we have also the ability to identify rare pathogenic variants, compound heterozygous and de novo variants, with also using some specific filterings for each use case. So now I will change my tab to show VarSome Clinical in more detail. Okay. Do you see my screen now? Okay.
Yes, we
Sure.
see. Yeah.
Thank you. Okay, so this is what you see when you enter into VarSome Clinical. Basically, here you have the different analyses. And here you have the different steps to launch the analysis. So the first thing would be to upload the files into the platform. Then you would define the samples. We will ask you for some specific parameters and some specific details depending on the use case, if it's germline or somatic. You can also input the phenotypes that the clinicians have seen in the patient. And we also have some filters that prioritize the variants based on these phenotypes. Then you would launch the analysis. You would select, if it's from FASTQ, VCF, the assay that has been selected, and you can also select specific filterings to be automated for when the analysis has been launched. And then you can also perform CNV analysis, both from FASTQ and VCF. So I have here an example of an analysis. Again, I need to switch the tabs. So here it is. So here we have an example of an analysis. This is an exome and CNV analysis. And the first thing I'd like to point out is the number of variants. So here you can see the number of variants. We have a high number of variants, and obviously it's practically impossible to go one by one manually reviewing these variants. So again, what we try to do is we try to provide specific filters to make your life easier to detect the variants of interest and to generate a report. So if we go from left to right, first of all, we classify the variants from more pathogenicity to least pathogenic. And if we go from left to right, we have the variant position, the variant type, gene symbol. We also have some specific manual classifications that can be created here. And every time specific variant appears, this will pop up and it will let you know who in your group classified it as, for example, in this case, pathogenic. And you can also have specific comments at the gene and variant level. Then we will let you know if there's an overlapping CNV with the variant, and also in the gene, the germline classification and the germline rules that have been applied based on the ACMG guidelines. Here we have more information about the variant, the overlapping genes, the mode of inheritance, the function, the zygosity. We also provide the phasing group for long-read analysis, the frequency. We provide the general population frequency, but you can also specify a specific ethnicity. And if you do so, we will show you the frequency based on that ethnicity. The allelic balance, the coverage. We also have some filters for the quality and the RSID. By the way, the columns can be modified and switched around depending on what you prefer to see first. This is quite relevant because depending on the use case, you would like to focus more on one thing than others. Apart from this, we also have, if you scroll down, many more information. First of all, I would like to start with the samples tab. Here we have the possibility to see in which other samples this specific variant has appeared. So you can see in all samples from all VarSome Clinical users how many times this variant has appeared. And then in your own samples, you will be able to see in which specific analysis, and you will be able to access them. And this will help you build your internal database the more you upload into VarSome Clinical. Apart from that, obviously, we have the germline classification. Here we show the classification and the germline rules that have been applied. And as I said, what VarSome does, it enables you to modify this if you have additional information. So you can activate and deactivate specific rules, and this will change the classification. Apart of that, if you have specific information that you would like also to include, you can also activate and modify specific rules. For example, if you have family history, you can also activate specific rules. Then we also have in silico predictors, the publications. We have either related to the gene or the variant level. And we have a very visual feature that you can use to filter out through specific publications. So here you can filter based on a specific term, or you can also filter from specific category. And here you have the different publications filtered also through year. So here you would click on this one. For example, if we filter through organ and tissue, we have here the eye. And here you will be able to access the publication. Apart from that, we have many other features. For example, we have the sample view. In this case, this is quite relevant because it will also enable you to see the different variants there is. And you can also check if there's a specific CNV that is overlapping with another variant. So you will be able to detect and to determine if it's an artifact or a true CNV. Here you can also see the different transcripts, and this is fully dynamic. So you can move around the different chromosomes to check this. Apart from this, as I said, we still have many variants here to determine and to analyze. So we have different filters that can be selected here if you click on create. And basically we enable our users to filter out through anything. So pathogenicity, mode of inheritance, specific chromosome, variant type, zygosity. You can also filter based on the phenotypes, etc. Here I have some specific filtering in which I filtered based on pathogenicity. So I would like only to include pathogenic and likely pathogenic variants, and then based on zygosity. This is obviously just an example, but it represents very visually how easy can the filtering be done. And you can see that basically applying these two filters, you can check there's only one variant that matches this specific criteria. You can be as stringent as you want with the filtering because you won't lose any variant. You can always go back here and modify the filtering, so you won't lose any variants during the analysis. Once you have the variants of interest, you can either generate a spreadsheet and download it, or you can also generate a report. So if you want to generate a report, you would go here to, first let me get, okay. You would go to generate a report. You can select it as primary finding and then click here and generate the report. And here you will be able to have you will see the basic information about the variant, and you can also input here all the linked information about the variant, including clinical trials, drug-related information, publications, reference, etc. And you just need to drop it into the report, and you will see it here. By the way, we also have the possibility to generate report templates in which you can include the institution logo. You can modify and have specific sections already determined. And once you launch the report, this will be automatically generated. So going back to the analysis, we also performed the CNV analysis. So here we have the CNV analysis in the other tab. To perform CNV analysis, we request our users to basically upload ideally between 5 and 10 samples from the same sex and sequencing run. And once you have the samples uploaded, you perform the CNV analysis, which is a sub-analysis. And the table, it's a very similar display, but with different information. So here we will show, again, based on pathogenicity, the different CNVs. And if we go from left to right, we have the position of the CNV, the length. Here we have the call quality, because as Jim said, there's specific limitations in in CNV analysis with NGS. So what we try to do is we try to provide the different call quality parameters to the users to determine if it's a true CNV or not. So we have the test sample coverage is sufficient in this case. If it has been if it has sufficient coverage and has been referenced with more than 2 samples, and if it doesn't fall into a camouflage region, which is specific regions in the genome that NGS analysis tools have difficulty calling the CNVs. Then we have the estimated copy number, the type of CNV, if it's a duplication or a deletion. You also have the possibility to perform the manual classifications. So you can also build your internal database with CNVs. The genes, the quality score. This one will tell you, so it will give you a value in which you can compare through the different CNVs that have been called, but you can't compare this specific value with other analyses. So it will give you a score of how likely this CNV call is reliable. Then we have the germline classification, the germline rules that have been applied. We have also included the possibility to see if there's an overlap with a known CNV. In this case, we see that there's an overlap with a known CNV. I will go into more detail for this specific CNV. Number of genes, number of exons, expected reads and observed, the read ratio. We also provide the frequency of the CNV if it's available, and also the cytobands. So if we check this specific CNV and we scroll down, we have again other features that we can check. So we'll have the publications that you can check that are related to this specific variant. We also have the CNV browser, which is one of the most interesting features for CNV. So let me check this. So here we have the, sorry, let me check this again. Okay. So here you will be able to see the different CNV calls and the different coverage regions that have been used for the CNV calling. And you can see that in the purple area, we see the expected range of the CNVs. And if it falls below, it marks it as a deletion. And if it's above, it marks it as a duplication. Here you can see the different coverage of the different CNV calls, and you can see you can also compare it to the reference samples. So here you can also switch to the different reference samples and check them directly without having to go back and forth with the other samples. So here you can see in more detail the coverage expected range. And in this case, these ones fall above the expected range. Apart from that, we have the CNV plot, which is a more static view, which can be used also to export the image into, for example, a specific publication. And we also have the known CNVs, which we can we let the users know if there's specific known CNVs in that specific region. Based on the different publications, data sources that we integrate. In this specific example, we have this CNV that has all of the call quality parameters that are correct. We have that is a deletion, and we see that there's an overlap with a known CNV. So this one would be one that we can focus on, and we could probably also include it in the report. So this is an overview of the platform. As you can see, what we try to do is we try to make it easier for our users to filter through the different variants that have in their samples, either for panels, exomes, or genomes. And once they have the variants of interest, they can generate a report to inform the patients. So yeah, I don't know if there's any specific questions. Thank you very much for your time.
Yeah, thank you very much for Andres. Hi everyone, if you have any question, you can open the mic micro and you can ask directly or type in the chat box, and we will receive your question and answer. Hi Andres, you have a question?
Hi, my name is Chu from the Illumina customer in Vietnam, and I have some questions for the panel. Your panel, is VarSome Clinical available for offline installations?
Sorry, for which solution?
Yes. Is VarSome Clinical available for offline installations?
Offline.
Here,
He mean the offline. He mean
yes.
the offline analysis.
Offline solutions?
Yeah, because our solution is online platform and based on cloud. His question is about, do we have and provide the offline analysis part?
Could I send my questions in the chat box?
Of course.
Yes, of course.
Yeah.
Yes.
And hi, Andres. You have a question about Nguyễn Hồng Thạnh. For CNV backbone spine panel, can we use the random enrichment pipeline for CNV variance calling as usual?
Yes, I believe so. Yeah.
And you can answer the question in the chat box. The second question is...
Oh, yes. So he mentioned if it's available for offline installation. So VarSome Clinical, it's web-based, so you need internet connection to perform the analysis and to log in. You don't have to install anything. It's web-based, so you just need access to internet. About the IVDR classification, that's a certificate that it's needed in Europe to comply with the standards of quality and security of the platform, and we have this certification. Then we also have a question. What is the meaning of a double X in the CNV column variant list windows? Does it mean that those CNV are coincident with a CNV? Okay, so let me go back to the example. I believe you're talking about the first analysis. Let me switch back the tab. So here, these two in the column CNV lets you know for this specific variant if there's a specific CNV that overlaps with that specific variant, and if there's a CNV in that same gene. So we show this for all of the different variants. For maybe there's a CNV that overlaps with this specific variant, and you are able to check in the sample view also here, and this can also help you determine if it's a true CNV or not. So here you can see if there's a structural variant that falls within the same region as the SNVs. Then we need ideally between 5 and 10 samples to perform CNV analysis, because we need to build the baseline for the coverage for the CNV analysis. Yes, you can also perform CNV analysis with from VCF. you just need to upload the VCF file, but we also support CNV analysis from VCF. Yeah. So yeah, I don't know if there's any other question.
Yes, you still have one question from Tees. Could you show us the guideline specification the VarSome Clinical follows to classify germline variants?
Yes. So I have, let me switch tabs here. Here we have the germline classification. And basically, here we explain the rules that have been applied, and we do like a scoring system. Each rule trigger is assigned to a number of points based on the strength of the evidence provided. So if it's supporting evidence 1 point, moderate 2 points, strong 4, and very strong 8. And then we have this total score, and we do this to classify it between pathogenic, likely pathogenic, VUS, etc. So here you have the possibility to modify this. Yes. Then Canvas and serve.
You can read the question and answer the question to let the customer follow that which question you answer for.
Of course, yes. So can VarSome serve as an alternative reference for unidentified variants in ClinVar? Yes. So we also, as I mentioned before, we have some specific variants that are novel, and we also perform the annotation of these specific variants. You can also check community contributions to see if there's any other user that has identified these specific variants. Does VarSome analysis accept the VCF file with HG38 reference genome build? Yes, we also support the HG38 reference genome, so no problem with that. What is the average support response time? So we have a great support team. This is one of our most valuable features of the platform. Usually we have a reply from our team within the same day. If it takes too long, maybe within 2 days, but usually it would be within the day or at least two days as much. Then if there's any discordance between ClinVar and VarSome, which database should be placed in priority for reference? So in this case, I don't know exactly for ClinVar and VarSome how the metric is done, but we have different strengths depending on the database. I don't know, Irene, if we have specific information for this one, but I will check it. But basically what we do is we take into consideration the different classifications from these databases, and we have a scoring system that taking all of this into consideration, we provide our general classification, our ICMG guidelines classification.
Excuse me, can you hear me? Yeah.
Yes.
I have just asked about the questions. I have to ask the questions about the discord between ClinVar and VarSome. Because in my practice there's two.
Đây ra bởi vì thật ra thì nó cũng bị cọc lại. Cái trang cọc tiếp đi.
Sorry. Because I met some kind of situation like there are variants reported in there are variants not reported in ClinVar, but only in VarSome, and there are some variants.
Từ chối ngay từ đầu làm và dược nó cũng bảo nói chung làm thì cũng do họ không hồi nợ đâu.
Alo, Khoa ơi, mute
Sorry,
ơi.
Khoa ơi, please mute.
Sorry. Can I continue
Sorry,
my question?
sorry. Yeah. Sorry.
Yeah. In my practice, there are two contexts. The first one is that there are some
Mày
variants.
nói chuyện đấu thầu nữa chứ.
There are some variants that are not reported in CleanVar but only reported in VarSome. And can we use like VarSome as references, alternative references in case of missing information in CleanVar? And in the second context, there are some kind of discordances between CleanVar and CleanVar classifications and VarSome classification. For example, in CleanVar, this variant was classified as like conflicting. For example, some reporter reported it as like pathogenic, likely pathogenic, and some others reported as like VUS. And in VarSome, I found that VarSome used like criteria from the AGMC, and based on these criteria, the classification was like pathogenic and likely pathogenic and even pathogenic. So in case there are like in case of discordant like that, which database should be used? For example, if this is conflicting or VUS, we don't like to treat the patients. We don't treat the patient based on the mutation result. But if it's like pathogenic, we will have another like treatment and interventions.
Yeah. So thank you very much for your question. It's a great question because sometimes, as you mentioned in CleanVar or in a specific other database, they provide specific classification. However, as we integrate many different other databases, we take all of this into consideration with different weights, and we can have discordance between what, for example, CleanVar says for a specific variant and what VarSome classified it. So yes, there can be some difference because we don't only classify the variants based on one specific database. We check different.
So actually, in my practice, I usually use CleanVar as the main reference, and in case there are some kind of discordance between CleanVar and VarSome, I would like to put this variant into a small like subcategory, for example, VUS with a higher tendency of pathogenicity, and then we will have a stricter follow-up for this patient. For example, VUS, we should check for the update of classification updates every 6 months for a year, for example. But for the variant with discordant like that, we should do like a more careful, stricter like follow-up for this patient, maybe 3 months or something, to see to hope that there will be any changes or updated in CleanVar database, of course.
Exactly. So there's also the possibility. Let me share my screen. Okay, I'm already sharing it. So there's a possibility to see the current annotation for a specific variant. So if there's a VUS that you want to check after a specific amount of time to see if it has changed, you can click here in current annotation, and it will automatically re-annotate it, and it will let you know the updated classification. So this is also a possibility that you can check from specific time to time if there's a change in the current annotation of the variant.
Okay, thank you very much. But actually, we just use the CleanVar free-to-use versions. We don't have like VarSome free-to-use versions, and we don't have VarSome Clinical. So technically, is there any difference between the VarSome Clinical and VarSome free-to-use relating to the classification? Actually, for example, is there any like update or something?
Exactly. So that's also the question that we have here from Tia. That's also a very good question, because we provide, there can also be discrepancies between what VarSome and VarSome Clinical show, because in VarSome Clinical we have additional data sources. So there's some commercial license that we need to pay and are included in VarSome Clinical. So these specific data sources that are not in varsome.com can also influence the classification.
Yeah, thank you very much.
Is there any other question?
I do see one question earlier with regards to the use of Illumina Dragon. Let me just read out the question. For CNV backbone spike-in panel, can we use Dragon enrichment pipeline for CNV variant calling as usual? So the answer is yes. I think the CNV spike-in panel, what it essentially does is it uses the spike-in probes to provide, that full genome coverage at regular intervals, and the Dragon enrichment pipeline can use that information. The only thing is we do need to have what we call a panel of normals to correct for bias using a reference. So Illumina has, a panel of normal already built for the Twist exome 2.5 panel. However, if we want to use the spike-in panel, then we need to create a custom panel of normal using, the recommended number of normal samples that is processed with the same spike-in panel, same library prep, same sequencing. So in short, yes, it is, the whole aim is to provide that regularly spaced coverage across the genome on top of exome coverage, and you can use other or your preferred analysis tools for that purpose as well.
So I guess there's no any other question.
Yeah, the question time still remaining. So do you have any question? If we don't have any question more, we will change into the Kahoot game, and we will have 5 questions and have 5 gifts for the correct answer. So when the winner, you answer the correct answer, please provide the phone number and email. We will send the gift because it is the card of the mobile phone. So we will directly to give you small gift via the phone. I will share my screen. Do you see my screen?
Yes.
Yeah. The question number 1 is what foundational technologies enables Twist Bioscience to synthesize millions probe with high precision? Dạ thì câu hỏi thứ nhất đó là công nghệ nền tảng nào cho phép Twist Bioscience tổng hợp hàng triệu probe với độ chính xác cao? Câu A là do độ tương đồng dữ liệu, câu B là do nền tảng silicon, và câu C là do phân mảnh cơ học và tổng hợp độ dài đoạn đọc. Thì không biết là mọi người có thể giơ tay và trả lời ạ. Rồi, bây giờ tại vì có ai giơ tay không ạ? Chắc là nhờ Dạ có ai giơ tay trả lời câu hỏi không ạ? Dạ có phần quà đấy ạ. Ai? Đâu? Chat hả? Lý thị, Su Phạm, Su Phạm B. Dạ chính xác ạ. Câu trả lời là câu B ạ. Ở đây thì em thấy có chị Lý Thị Thanh Hà và bạn Su Phạm thì trả lời là câu B. Dạ đúng, câu B là đáp án đúng ạ. Thì em cũng xin anh chị cho lại số điện thoại để mà tụi em có thể chuyển cái phần quà trực tiếp sang số điện thoại của các anh chị luôn ạ. Dạ thì do là khi mà em xem màn hình đó thì em sẽ không có thấy được cái câu trả lời, nên là chắc là nhờ anh Tùng hoặc là chị Kiều hoặc là một thành viên của AI có thể list cái phần chat ở trong cái meeting bot hoặc là mấy anh chị có thể mở mic để mà mấy anh chị giơ tay trả lời ạ. Dạ em xin tiếp tục cái question number 2 ạ. The question number 2 is whole exome sequencing focus on the protein coding portion of the genome. Approximately the percentage of the genome does this represent? Thì các giải trình tự toàn bộ vùng exome tập trung vào vùng mã hóa protein của hệ gen, thì phần này chiếm khoảng bao nhiêu phần trăm của hệ gen? Câu A là 1%, câu B là 5%, câu C là 15% và câu D là 50% ạ. Nguyễn Tiến Đạt câu mấy? Còn ai nữa không? Nguyễn Tiến Đạt câu A. Dạ em sẽ lấy cái người nhanh nhất đầu tiên ạ. Dạ đúng là câu A là câu trả lời đúng ạ. 1% ạ. Người trả lời chính xác và đầu tiên xin cho em lại cái số điện thoại đi ạ. Và câu hỏi thứ 3 là why is detecting copy number variation CNV challenging when using standard one exome sequencing? Thì tại sao việc phát hiện biến thể số bản sao CNV lại khó khăn khi sử dụng giải trình tự toàn bộ exome tiêu chuẩn? Câu A là exome probe degrades too quickly. Các probe exome thì bị phân hủy quá nhanh. Câu B là there are gaps in coverage between the gene coding region. Thì có các khoảng trống gap trong cái độ bao phủ giữa các vùng gen mã hóa. Và câu C là CNV only occurs in RNA changes. CNV chỉ xảy ra trong các bản sao RNA. Và câu D là microarray sequence the gap better, là microarray giải trình tự các vùng gap tốt hơn. Thì Nguyễn Hồng Thanh câu B. Dạ vâng, chúc mừng anh Thanh, câu B là câu trả lời đúng ạ. Anh Thanh cũng cho em xin lại số điện thoại đi ạ. Và câu hỏi số 4 là what's our one advantage of using VarSome for variant interpretation? Thì lợi ích của việc sử dụng VarSome trong việc giải biến thể là gì? Câu A là thay thế hoàn toàn các thí nghiệm trong phòng lab. Câu B là tổng hợp, gom lại tất cả các bằng chứng vào một nền tảng duy nhất. Câu C là chỉ hiển thị dữ liệu thô. Và câu D là chỉ hoạt động offline ạ. Có câu trả lời chưa ạ? Nguyễn Hồng Thanh thì chắc là được rồi nên là bây giờ nhường lại cho Mãnh Hùng Nguyễn anh Hùng ạ. Câu trả lời câu gì? Câu B. Dạ đúng ạ, câu B là câu trả lời chính xác ạ. Chúc mừng anh Hùng ạ. Anh Thanh cũng trả lời đúng, nhưng mà do anh Thanh đã trả lời trúng câu trên rồi nên là em xin phép được nhường phần quà cho anh Hùng ạ. Và câu hỏi cuối cùng là VarSome tích hợp dữ liệu từ nguồn nào? Câu A là only user upload file, là chỉ các file do cái người dùng tải lên. Câu B là multiple genomics database and prediction tools, nhiều cơ sở dữ liệu gen và các công cụ dự toán. Câu C là only hospital records, chỉ từ hồ sơ bệnh viện. Và câu D là social media platform, là từ các nền tảng mạng xã hội ạ. Câu gì? Câu gì? Câu B. Dạ vâng, câu B là câu trả lời chính xác ạ. Dạ. Dạ, em không biết là mọi người win trong trò chơi này nhanh nhất là đã cung cấp lại cho tụi em số điện thoại hết rồi phải không ạ? Chị Su Phạm, chị Anh Hùng, Thị Hà, chị Linh. Hình như em thấy là còn một vài Như Phạm đúng không? Dạ em dạ phần quà là một cái voucher card điện thoại 100.000 ạ. Nên là tụi em sẽ chuyển khoản vào các bạn đã thắng trong cái trò chơi lần này. Thì chân thành cảm ơn tất cả các quý anh chị đã dành thời gian để mà tham gia cùng cái buổi hội thảo online webinar ngày hôm nay. Thì cũng mong rằng cái nội dung của buổi hội thảo cũng giúp được các anh chị trong cái quá trình nghiên cứu ạ. Hi Jim and Andres, thank you for your participation.
Chị Vân ơi.
Chị nghe.
Anh Tú muốn hỏi thêm câu hỏi ấy chị.
Đâu, anh Tùng muốn hỏi thêm câu hỏi ạ? Anh Tú, please ask the question. Đâu, anh Tú câu hỏi chỗ nào đâu thôi. Hi, Andres and Jim, have one more question for you. So we should wait in. Mr. Tu asks, is Twist CNV spy-in panel compatible with the SM2.5 from Illumina and other West panel?
Yes, technically it would be compatible. I wouldn't be able to say so for other WES panel because that spy-in panel is specifically designed to cover the gaps in the Illumina, not Illumina, in the Twist exome, right? And the Twist exome. And in fact, Twist supplies Illumina with the exome panel, right? So this is something that Illumina also mentioned, right, in their website as well. So in that case, with the Illumina exome, it would be compatible. But for other WES panel, that may not be the case because the specific gaps in the other panel will be different than what we designed. So I just want to add that besides the spy-in panel, we also provide what we call a whole genome CNV panel. So this would be independent of any exome panel spy-in in the cases where sometimes we want to design a custom panel and we want to have some CNV detection capability either at specific chromosome or the whole genome. That is where we can actually kind of select a certain number of the probes for this CNV detection that is independent of exonic content. Yeah, hopefully that addressed the question. Basically, yes, for Illumina, yes. For this other WS panel, probably no. And if in the event that we are using a non-WES solution, that we have other kind of CNV spike-in solution available as well. Right.
Yes, thank you. Very clear clarification. Yeah. So thank you Jim, thank you Andres. Yeah, em cũng xin cảm ơn tất cả mọi người ạ. Yeah. Now we can end the event. Thank you.
Thank you very much.
Bye.
Yeah.
Có quà cái thôi nói liền ha.